Hi, myself Zubairuddin But welcome to my another topic, that is topic number 12.6, that's mechanochemical systems. Muscular contraction is the most thoroughly studied mechanochemical phenomena. To this category belong a number of numerous biological processes, the movement of plants, the movement of cells with flagella and cilia, and the entire set of the motion in mitosis and meiosis, the movement of protoplasm inside an undividing cell, contractile processes in the tail of phage particles, mechanochemical processes in the membrane, the movement of ribosome related to mRNA in polysomes, mechanoreceptions. Flagella and cilia are the special contractile systems of bacteria and protozoa that provides their motion in a liquid media. The cilia function in a number of organs of multicellular organism. For example, comb bears seen gooseberries regulates the orientation of their bodies by the coordination motion of cilia. The cilia creates a liquid current in the gills of bivalves in the human trachea, etc. Flagella and cilia have similar structures. They are extended slender filamentous structures with a length ranging from several micrometers to several millimeters, with a length millimeters and a diameter varying from zero point 0.1 to zero point 0.5 micrometers. Electron microscopy shows the flagellum is a bundle of 11 thin fibers consisting of a circle of 9 double figures around the central single microtubule, that's 9 plus 2 arrangement system that have a fibril structures. The diameter of the central fibers and the flagellum is about 24 nanometer, and the distance between their centers is 30 nanometer. The lateral size of each of the nine doublets peripheral fibrils is 37 into 25 nanometers square. The protein flagella and cilia have been studied a little. Their ATPase activity has been established. The forms of frequencies and the sine wave beat propagating along the flagellum from the base of the contractile, but they differ from myosin and actin. The model theories of the beating of flagella and cilia are concerned with changes in the conformational state of protein contractile units when a contractile wave reaching them. In each unit, there occurs the splitting of ATP. In flagella and cilia, there is sliding between subfibrils and the arm, functioning like the bridges in the actinomycin of the muscle. No detailed molecular theory according for the wave-like beating of flagella and cilia has yet been worked out. The contractile protein has also been identified in other systems. Thus, it has seemed until recently that the wheel has devised by the human mind alone during the natural evolution. There could not have or or seen a microscopic device for rotation about the axis. It has been found, however, that there is something like the wheel even in the bacterium E. coli. Each cell of E. coli has four long flagella. Their rotational motion according to the motility of the cells. In the basal body of flagella, which resides on the cell wall and membrane, there is a wheel, a ring composed of 16 protein molecules in the membrane, opposing a similar ring in the cell wall. Uh, Shown in the figure, scheme of the locomotive motive device of E. coli is clearly mentioned in a figure, that's figure 12.21. The plasmodium of myxomycetes perform active oscillatory motions of the protoplasm. It contains actin and myosin, which are similar to those of the muscle. The myosin of the plasmodium, however, does not thick filaments. Instead, it forms only small oligomers. Nevertheless, the myosin interacts with the actin of the plasmodium. In the plasmodium, there have been detected thin bundles of filament composed of actin and myosin. These bundles appears and disappears following the oscillatory phases. The protoplasm current is regulated by calcium 2 positive ions, probably with the uh, participation of regulatory proteins that have not yet been identified. The motility of the protoplasm might be expected to be similar to the movement of the muscle, but the absence of thick filament defies this similarity. So there is every ground to believe the mechanoreception and also sound reception are associated with mechanochemical processes. It is possible that mechanical reception is tied up with the mechanical effects of ion channels in the membrane of receptor cells. With the deformation of these channels, the ion channel may be considered to be vectorial enzymes. They transform the incoming ion or substrate to the outcoming ion, that's the product. A single mechanoreceptor, the Pacinian body, is shown schematically in figure 12.22. The receptor consists of nerve ending bounded by a capsule made up of peripheral and a central zone. The peripheral zone consists of about 30 closed elastic sheaths. To the central zone contains 60 tightly packed cytoplasmic layers separated by clipped oriented along the long axis of the elliptic cross section of the nerve ending. The modulus of elasticity of the sheath of the peripheral zone is about 10 power 6 grams per centimeter cube, that's 100 Pascal. The sheath are capable of secreting in a mechanical force within the range of 150 to 200 dynas. Thus, the most general proposition of biological mechanochemistry is its obligate relationship with the enzyme activity of working substrate, contractile and regulatory proteins. Contractile proteins, notably actin, are contained in a number of other cells and tissues. Actin constitutes about 20% of the entire protein in the neurons of a chicken. Actin or actin-like proteins is present in the embryonic lens, lungs, skin, heart and pancreas, kidneys and in the brain tissue of the chicken. Actin or myosin-like proteins have been isolated from thrombocytin and form a complex contractile protein in the human blood platelets, thrombocytes. These factors confirm the general proposition concerning the necessity of mechanical motion in life processes and the similarity between the mechanism of this motion in the large variety of biological systems. Thank you. This is all from this topic. Have a nice day.